Welcome to STEM Talk.
STEM Talk. STEM Talk. STEM Talk. STEM Talk. STEM Talk.
Welcome to STEM Talk, where we introduce you to fascinating people who passionately inhabit the scientific and technical frontiers of our society.
Hi, I'm your host, Don Cornegas, and joining me to introduce today's podcast is the man behind the curtain, Dr. Ken Ford, IHMC's director and chairman of the double secret selection committee that selects all the guests who appear on STEM Talk.
Hello, Don. Great to be here.
So our guest today is Dr. Euan Ashley, who is a leading pioneer in the use of genomic sequencing to solve some of the most puzzling medical mysteries that people face today. Euan is a Scottish-born professor of medicine and genetics at Stanford University, and he's also the author of the Genome Odyssey, Medical Mysteries and the Incredible Quest to Solve Them.
Before we get to our interview with Euan, we have some housekeeping to take care of. First, we really appreciate all of you who have subscribed to STEM Talk, and we are especially appreciative of all the wonderful 5-star reviews.
As always, the Double Secret Selection Committee has been continually and carefully reviewing iTunes, Google, Stitcher, and other podcast apps for the wittiest and most lavishly praise-filled reviews to read on STEM Talk. If you hear your review read on STEM Talk, just contact us at stemtalk@ihmc.us to claim your official STEM Talk t-shirt.
Today, our winning review was posted by someone who goes by the moniker JCJ. The review reads, "The other day, I read an article about a new study that showed how diet in combination with exercise has the ability to transform our bodies and health. We so often focus on diet in terms of losing weight, but this study emphasized the importance of building muscle to lose weight. But I already knew that. I knew that because I listened to the Donald Layman interview you guys had, and that's what I love about STEM Talk. I feel as if I'm ahead of the game and the latest research."
Well, thank you so much, JCJ, and thanks to all of our other STEM Talk listeners who've helped STEM Talk become such a great success.
Okay, and now on to today's interview with Dr. Euan Ashley.
STEM Talk. STEM Talk. STEM Talk. STEM Talk.
Hi, welcome to STEM Talk. I'm your host, Don Cornegas, and joining us today is Euan Ashley. Euan, welcome to the podcast.
Oh, thanks so much for having me.
Yeah, and also with us is Dr. Ken Ford.
Hello, Don, and hello, Euan.
Hi.
So, Euan, let's just get started. I understand that you were a computer nerd growing up and that your fascination with science fiction played a role in that developing interest. Is that correct?
Yeah, I was born a nerd, trained in a few different ways after that, but definitely a nerd growing up. I loved any and all gadgets. I loved computers as they were coming online. I grabbed the first, Game Boy, which came many years later, but the first handheld gaming device. I had it in my hands, had to be wrenched free. Anything computers, I was definitely ready to ingest and hold on to as a little kid.
So on that question, how were you first exposed to computers and what was it about computers that hooked you?
Well, we lived in the UK, and some of the computers I think that were available there were also available in other parts of the world, in the US in particular. But the computer that we had at home was called the ZX Spectrum. It was a small computer. I think it had maybe 48K of memory, had rubber keys, and you could program in BASIC, or you could play games. And I did both of those things as a little kid and just immediately found an affinity with this, whether it was the addictive nature of the games or just being able to make a machine do something that you wanted it to do. It just seemed magical to me, and I just couldn't stop.
And when you were a teenager, I hear that you worked on developing smart tax software for your father. So how did that turn out?
Yeah. I really was a nerd, wasn't I? Yeah. So, yeah, I like to program little things, and most of them were relatively pointless, just to demonstrate that something could be done. And I think my dad spotted that and maybe thought that this could be channeled into a slightly more positive direction. And he used to prepare the taxes for his medical practice. He was a GP. And he had every year had to take out these books of tables and then look up for each employee, page 14 and column Y and, row X. And it just, I spotted that there was a pattern, basically. It wasn't too difficult to see, that could be expressed as a mathematical function, and suggested that maybe I could build that in so he didn't have to look up these tables. And somehow, I guess, I just spent the summer doing that. And by the end of the summer, we actually had a working version of the tax program. So maybe I should have spent a little bit more time outside. But I think when you get into something, you just want to get it done. So it was, that was probably the first complete end-to-end piece of software I ever wrote.
That's pretty cool. I imagine you also tried your hand at developing computer games, or at least playing around with the thought of developing them. Is that right?
How did you guess? Yeah, yes, indeed. Indeed. So my friends were very much into computer games. They did a bit of coding, but mostly they played games. And so I was trying to find something to amuse them with. There was a game that was very popular on the ZX Spectrum based on the celebrity who was Daley Thompson, who was one of the world's best decathletes. And basically you got to move your little Daley Thompson through 10 different events. And most of the events, basically, you made your competitor go faster by alternately pressing these rubber keys, like as fast as you possibly could. I mean, in a really interesting twist, I later learned that the rapidity with which you can alternate muscles back and forth across the same joint is actually related to sprint performance. So that's like many years in the future I was to learn that. But at the time, there was all these kids like bashing the keys of this rubber keyboard to make their little Daley Thompson run faster or leap higher. And that became the inspiration for my game. I thought, well, there's a way we can have an input. What about, why don't we do horse racing? And at the time, you basically, to do graphics, you would have to basically build a grid and then make a little horse inside the grid. I think I had several grids for the horses in different positions of running. And then basically 2 people would compete against each other and the computer to make their horse run as fast as possible along the course. And we had some random speed elements for the other horses in the course. So yeah, that was another, it did not make me a fortune, that game, but I think my friends thought I was reasonably cool, maybe, but certainly was fun to try my hand at making a computer game.
So Euan, let's back up for a minute. Where did you grow up?
Yeah, you may be able to tell from my accent that it was not in California or at Stanford, where I now live, but rather in Scotland. So in the west coast of Scotland, in Glasgow, actually.
And so your father is a physician and your mother is a midwife. And I understand that early on you would tell people that you wanted to be a physician, even though your parents didn't necessarily push you in that direction. So what was behind the drive of yours to become a physician?
Yeah, it's a great question. It's just something that appeared to be deeply in my soul. I can't think of any other answer. I mean, from the youngest age I can remember, which is probably 4 to 5 years old, when any adult would ask me what I wanted to be when I grew up, I would answer that I wanted to be a doctor. I just felt like I was put on this earth to try and help other people through medicine. And I never considered anything else, just from an early age. I think many would assume that it was the influence of my mom and dad, both in the medical profession. And I think that has to be at some level the case. But interestingly, they both at one point or another, more on my dad's side, if anything, pushed me a little away from a career in medicine. But despite that, it was never going to be any other way for me.
And at some point, you became quite fascinated by data and statistics. How did this interest come about? What motivated or sparked this interest?
Yeah, I know numbers always seem to make sense to me and seem to be fascinating, and the patterns that you could see within numbers, whether it was prime numbers or the way that numbers related to the real world, just always seemed fascinating. I would memorize numbers much more easily than I would memorize words, and they seemed to almost sing to me in a way that just made sense. And so I think that combined with my computer geekery, which was somewhat mathematical as well, to give me this sense of the world as a mathematical place. And at school, even though, again, I was always telling everybody that I was interested in medicine and science, my teachers were saying, but the things you seem to be good at are maths and physics. And are you sure you want to do biological science? Because you're just moderately okay at that. And I would be like unpersuaded. I'm like, no, you're right. But I definitely want to do medicine. And it was much later, to be honest, that I realized that it might be possible to combine the two. I really had always these two parts of my brain. One side I was clear I wanted to be a doctor, and on the other, I was in my spare time a computer geek. But I honestly would never have thought that it would be possible to combine those two interests.
So you graduated with first-class honors in physiology and medicine from the University of Glasgow. And then you headed off for your medical residency and a PhD at the University of Oxford. And so somewhere along the way, you met a fellow student named Fiona. So how did you meet Fiona, first of all, who I understand helped you through medical school and has played a pretty major role in your life? Is that correct?
That is correct. And it is about my wife, about whom you speak. I met Fiona in Glasgow, and she was also a scientist at the time. I remind her of this from time to time, or she reminds me occasionally that she was doing RT-PCR long before I was ever doing that. She was actually a microbiologist for her undergrad, and so we met. But we actually met through kind of sport and exercise, and she later became the president of the University Athletic Union at Glasgow. So she stayed around a little longer after her undergrad while I was in medical school, and our relationship flourished. And that was quite some time ago now, almost 30 years ago. But yeah, she's just been an incredible support. We ended up moving to Oxford at the same time, she for a job and me to go and do residency and then a PhD. And then we came up with this crazy plan to move to California. And that's where another story began.
Well, as you just mentioned, the two of you took off for California, and that was for you to conduct your postdoc research at Stanford. It must have been a good fit. You must have liked it there because you ended up joining the faculty in 2006, and we'll come back to that. But what brought you to Stanford in the first place? What motivated you? Said you had this crazy idea and you both moved to California. What was the driver there?
Yeah, I was lucky enough actually as a medical student to get a connection to California. You know, the medical students are often recommended to try to do an elective during the summer period, go somewhere else in the world and just experience medicine in a little different environment. And I went to one of the cardiologists. I was already interested in the heart. So I went to one of the cardiologists in Glasgow and asked, who do you know in the world, outside of these walls? And he gave me 2 or 3 names and I wrote to each one of them. And they each wrote back, actually, amazingly, and said, sure, if you want to come, you'd be welcome to. But one note in particular really stood out, and it was from a professor called Vic Frolicher here at Stanford, cardiologist, world expert in exercise and the heart, literally wrote the book on that. And he wrote just such a cool note back to me that had humor and encouragement and saying that, he might be able to help out with somewhere to stay and maybe I could use his bike. And it just seemed like a wondrous opportunity. And it actually became now a 30-year-long friendship. So as a medical student, I reached out and came to California, and I was just blown away by Stanford. Just blown away. It was the summer that Netscape was floating. So it was actually the beginning of the internet boom. The internet was still a new thing. Bill Gates hadn't added the addendum to his own book about how the internet's going to be important. So it was pretty far back. I came from the west of Scotland, which has many great things about it, but weather is not one of them. And so I came from a rather rainy place to just beautiful weather. I had this bike that I could cycle around. And where was I cycling? To get to work in the morning, I would cycle through Xerox PARC, where they invented the mouse and they invented the graphic user operating system. And it was in geek heaven. And as well as that, I got to study data relating to exercise in the heart. So I was just absolutely in heaven. And at that moment, really, I'd said, if there was any chance of coming back to this incredible environment at Stanford, I would absolutely take it and not worry about the consequences. So that's what took us there a little bit later, where I came out for a postdoc.
So I'm curious, what was it about the heart that fascinated you? And at what point in your studies did you decide to specialize in cardiology?
Well, I think coming from the west of Scotland, heart disease is something that is very much around you. And it was in my family, and it was in the many friends and relatives that one point during medical school, one of the lecturers basically put up a series of slides that identified the place in the world with the highest rate of heart disease. And they started with Europe, and then Britain, and then Scotland, and then the west of Scotland. And then literally it was outside our door where heart disease had, at that moment in time at least, the highest prevalence. So that was an important part of it. But actually, if I really think about what drew me into cardiology, it was a singular moment in the physiology lab as a second year student. And we basically had this opportunity to look after a Langendorf beating heart. So Langendorf was a scientist who essentially developed a number of really important physiological experiments. But one of them was the isolated heart, where you could take from an animal a heart and hang it essentially through the aorta, the big blood vessel comes out the top, and then feed it sugar, feed it energy, feed it oxygen, and keep it alive. And the incredible thing was that this heart just kept beating, and it would beat for hours. And so we came into our physiology lab, we sat on these wooden stools and sort of rickety old lab from, for all I know, hundreds of years ago at the University of Glasgow. And suddenly we had this just beating machine, this sort of symphony of biology in front of us, 4 chambers beating kind of in unison. And as long as we kept feeding it, it kept beating. And I stared at that thing for hours and hours. And I said to myself, I have to understand how that organ works. It just seemed like the most interesting organ in the body. It's the only one in continuous motion, 3 billion beats in your lifetime. It creates music with its sounds as the valves open and close. It has rhythm. It just seemed like how could anyone not want to spend their lives devoted to understanding that organ? So I think that was the day that the seed was planted.
Really interesting and well spoken, Euan. In terms of your professional career, you started out with a clear focus on medicine, as we've discussed. And also, as we've discussed, you had this extensive interest in computing and data. At what point did you realize these two really would combine beautifully to afford you an especially interesting career opportunity?
Yeah, I think that was really the first time I came as a postdoc to Stanford. So after we jumped ship and came across the Atlantic and landed at Stanford as a postdoc, I came to the lab of Tom Quitermis, who's the chief of cardiology here at Stanford. And it was the early days of the microarray. So microarray, of course, Pat Brown and others developed this technology in the early 2000s to go from taking molecular biology from thinking about one gene at a time, essentially maybe one protein at a time, to a higher throughput entity. And at first that was a higher throughput was maybe looking at 20 at a time or 50 and then 200. And over the course of the time after I joined the lab, we were moving from a time where you could suddenly measure 2,000 gene expression of 2,000 genes up to the full genomic complement of genes up to 20,000. And this seemed to me just kind of mind-blowing that you could measure the gene expression of every gene in the genome. But it was just the beginning. But what it also was an opportunity, because suddenly there were molecular biologists who were kind of used to their experiments with an N of 3 or 5 or 10, and they were ready with their statistics to do that. But suddenly they had a data file with 20,000 data points times however many different settings that you're measuring them in. And you just need a whole different set of tools to do that. So I think I definitely have to credit Tom Quitermis. I was new in the lab and he needed somebody perhaps with some geekery to take on the concept of thinking about 20,000 measurements. And so I sought out some statistical mentors at Stanford. And I think that moment the die was cast. And it really became clear to me that these things could be combined and that it could be quite useful to be able to make computers do things in a world, in a molecular world, where you were making many, many thousands of measurements. And of course, we moved from there to millions and then billions of measurements. And so data science, biomedical data science sort of became born.
Just as an aside, I did my PhD at Duke University, and my thesis was actually focused on gene expression microarray and translation of it. This is back in 2008 when I started, and translation of it as a diagnostic tool. And so I joked around that my PhD became less about biomedical research and more about biostatistics and data science because of the reasons you're talking about. So I totally understand.
Really exciting time.
Yeah, absolutely. So currently at Stanford, your lab is focused on the science of precision medicine, and you're best known for helping establish the field of medical genomics. And so you and your colleagues at your lab developed some of the earliest tools for the interpretation of the human genome in the context of human health. So can you give our listeners just a short primer on the genome and then also how the first draft of the human genome sequence was completed? That was about 20 years ago.
Yeah, of course. So the Human Genome Project really was like landing a human on the moon. It was a true moonshot, and I'm happy to say we're still in a world where we make attempts on those. But this was, a project that was funded for $3 billion. There were 10 countries involved. It took basically 10 years to get to the first draft of the human genome. And when we say human genome, really, well, we each have 2 copies. We get a chromosome each from mom and dad. In reality, the human genome was really just one. And so we call it monoploid. So it's just one long line of letters, essentially, for each chromosome. And initially, the idea was that would be a mixture of 10 people's DNA, so that nobody would know whose DNA was in the human reference at any given point. But for a variety of technical reasons that only became clear later on, it actually turns out that it's mostly one person and who is anonymous and one person who was actually one of the scientists connected to making those libraries at the time. Either way, this human genome reference has become the single kind of most important contribution, I think, maybe to biology, but certainly obviously foundational to the field of human genomics, because every genomic experiment that came afterwards, whether it was gene expression experiment or human genome sequencing or genome-wide association study in a population cohort, has relied to some extent on that human genome reference. But yeah, it was several individuals from Buffalo, New York, who answered an ad, and they came along to have their blood taken, actually by a set of identical twins, which I thought was a really nice sort of genetic element to the beginning of the Human Genome Project.
Nice touch.
Yeah, exactly. I like that. So that's where we are. And actually, the sequence was arguably completed last year with a project called the Telomere to Telomere Project. It was a draft in 2002. It had been improved over the course of the last 20 years, but it took a huge leap forward last year with the work of the Telomere to Telomere Consortium, who actually added 8% that had been missing, mostly around the telomeres, the caps of the chromosomes. And so we really are very close, first of all, to one fully finished genome. And much more important than that, and I think more exciting even, is another related program called the Genome Project, where we start to look at the diversity across the globe, of course, and looking at multiple genomes. And so that's really been the trajectory for genome references. At some point with the current technology, we might actually be able to leave the reference aside and just think about every new person, every new genome that comes along as its own set of data. But at the moment, we compare every new genome to the reference genome.
You often talk about genomic medicine and precision medicine. For our listeners, could you briefly describe what they entail in this context?
Yeah, of course. And I think what the Human Genome Project really opened up everybody's eyes to the possibility of understanding the genome at a deeper level, understanding our biology, understanding maybe at some level what it means to be human. And of course, the first step in that process was to use that to fuel discovery. But very shortly, a few short steps beyond that is the application of that genome to medicine. So we think about being able to diagnose rare disease, or being able to give people better risk prediction for the future, or helping them understand which medications would be more suited to them in relation to their particular genetic makeup, something called pharmacogenomics. So when we talk about genomic medicine, we really talk about something in that range. And the genomic medicine really seeded precision medicine, though precision medicine, much broader term. You may have also heard of the term personalized medicine, and personalized medicine really encompasses the ideas I mentioned just a moment ago for genomic medicine. But precision medicine was really a way of really thinking about disease at a deeper level and kind of redefining the map of diseases. And really, as we understood a disease at a molecular level, it might turn out to be 5 different diseases. And it might turn out that each one of those 5 groups should be treated differently. And if we do find medications that do that, we find much bigger effects. And that has really come to pass. But it was really President Obama that rocketed the term precision medicine to the top of Google search terms. If you look on the Google Trends tool, you can see that just before the State of the Union, when he announced the Precision Medicine Project, there were very few searches for precision medicine. But shortly after he announced what was coming, several $1 billion of funding, of course, everybody switched out the word personalized for precision in their slide decks, me included. But there are, of course, some overlapping elements. But the idea of futuristic medicine, where you get the right medicine to the right patient at the right time, that's the essence of precision medicine.
And so I understand that the aha moment came for you back in 2009 when you walked into the office of a friend who was just the 5th person in the world to have his genome sequenced. So can you share with listeners the significance of this and also the impact that it had on your life and your work?
Yeah, it was a really pivotal moment. And I think you walk into these moments not having any idea what they are at the time, but it's only in retrospect you realize just how important they were. I was going for a meeting with someone I didn't know that well at the time, actually. He became a friend later, colleague and then friend. But Steve Quake was the chair of bioengineering here at Stanford. And he made a name for himself already and started multiple companies and written a lot of really important papers around microfluidics. He was an inventor, really a genius character around campus. And I was going to his office for a very simple reason, which was that we had a new chair of genetics coming, Mike Snyder, and we needed to organize a seminar, kind of welcome him to Stanford. So Steve and I were going to organize some of the geneticists on campus for that purpose. So I came, I've had to find his office as usual. It's impossible to find in his labyrinthine Clark building here at Stanford. And then I found it, and here was him in this office. And you can imagine piles, and these days people don't have this, but piles of journals, like printed journals, like almost to the ceiling, at least in my head, it was to the ceiling. And so I had to sort of find my way through that to find him in the corner where he was pecking away at a keyboard. And so I said, can we chat about this seminar? He said, okay, but first, here, come and take a look at this. And he starts pointing at the screen at what looked like one of those old tables, HTML tables that used to be on old website. And it said trait-o-matic at the top. And then there were lists of genes and lists of variants. And, I hadn't really seen anything like this before. And I was saying to him, well, what is that? And he said, well, that's my genome. And as you said, he was like the fifth person in the world to have sequenced a genome, which he did in his own lab with 2 people for a tenth of the price of the one before that. And so this was just mind-boggling. My jaw was on the floor. This is somebody's genome I can browse on the screen right in front of me. And just to put this in context, we were sending genetic testing from our clinic at the time. But we would send a test for 3 genes. So you'd get only the genes, nothing in between. And it would take 3 months and cost like $5,000 to $10,000. And he was literally showing me a browser for an entire genome, 20,000 genes, plus everything in between. Remember, the genes are only 2% of the genome. And the whole thing was there. It was mind-boggling. But then another thing happened because he started pointing. He said, "Hey, look at this variant that I have. Well, look at this one. Have you ever seen that gene before?" First of all, did he think I had some photographic memory for every gene in the genome? I mean, I did not. But he actually started pointing at some genes that I did know that cause rare heart diseases, and in fact, rare heart diseases that can be associated with sudden cardiac death, very severe conditions. And he appeared to have genetic variants in them. And so suddenly I was asking a few more questions, and actually the whole session changed from being a scientific discussion among colleagues to almost like a medical examination. And so I started asking about his family history of heart disease. And at first, of course, like most people, he said, "No, I don't have too much of that." But then as I pushed him a little further, it became clear his dad had a cardiac arrhythmia. There were other grandparents in the family who had early heart disease, many individuals. And then finally I asked him, has anyone ever died suddenly or unexpectedly, which is really our single most important question to help discover these conditions. And he said, "Well, it's funny you should ask, because my cousin's son, who was a 19-year-old, no previous history, just didn't wake up one morning, died sudden cardiac death with no known cause." And so we moved all of a sudden from this, incredible discussion about his genome to this really tragic discussion about his family, the family member, his cousin's son, who had died. But also in that moment, it became clear that we had this unbelievable opportunity, because he was for sure coming to my clinic, with this family history. And he said, "Oh yeah, my family have been telling me I need to go see a cardiologist." And I'm like, "Okay, well, there's one right here." And as I invited him to my clinic, it suddenly dawned on, I think, both of us that he was about to become the first patient really in the world to walk into a clinic with his entire genome available to the medical team. And after that sank in, it's like panic set in, because what were we going to do? How were we going to analyze an entire genome? This is 6 billion data points. As I mentioned, we're in cardiology, we're used to like a lipid panel, like a cholesterol panel with 5 numbers. And suddenly we were going to have 6 billion. And so that was the beginning of an adventure that initially took us, 6 to 12 months. And that in reality has been 15 years now. It became an amazing friendship with Steve, many scientific collaborations. But yeah, it's not often you know your friends as deeply as we know Steve, all the way down to the last base pair of his genome.
So yeah, on that note, you actually wrote a paper about Steve in 2010, where you described how you put together a team to undertake an integrated analysis of a complete human genome in a clinical context. And this is a groundbreaking paper because you were asking the question of how you bring together the entirety of genetic literature and everything that is known about associations between genes and disease and the variants of diseases. And so you built all these questions into an algorithm that could be deployed in the context of a single patient in a primary care practice. And I'm guessing that was no small feat, was it?
It was kind of a crazy undertaking, but some of the most fun things in life come from slightly crazy ideas. And I often think about that TV show, Who Wants to Be a Millionaire, where you have this opportunity when you get a hard question to call a friend. And that was kind of how I felt there. Clearly, Steve was going to be coming to our clinic. I felt very comfortable taking care of the cardiology elements, and certain elements of the analysis of his genome were already starting to form in our minds. But there's no way you could take on something this big without a lot of friends. So I started calling some individuals. Atul Butte, who's now at University of California, San Francisco, was at Stanford at the time, and he had built some tools for thinking about common disease and common genetic variation. Russ Altman, still here at Stanford, had been building for a decade or more the PharmGKB, the pharmacogenomics knowledge base, which remains the kind of Bible to this day for pharmacogenomics. And so they were two of my first calls. It was very clear we'd need to think carefully about the ethical implications of what we were doing. So I called Hank Greeley, who's a world-renowned ethicist, particularly around medical issues and especially technology and medical issues. And so we formed this team to really try and start to think about this. And then, of course, the people who had helped analyze, at least with that first table that I saw in Steve's office, were from Harvard. Actually, George Church's group and the Tratomatic software had come from there. And so the Personal Genome Project folks joined the project too. And we just basically everyone put down all their other projects basically. And we did this one thing together for, I think, 9 months running at 100 miles an hour, many, many late nights to try and see if we could interpret one person's genome in the context of the entire medical literature on genomics, which was, ridiculously ambitious in retrospect. And we only made a small bite. But I think, as I always say to my team, you can't do two until you've done one. And so you just have to focus on the first one, do the best job we can. And, I think in the end, we made a first step, and it was a pretty exciting time.
I think it was a really neat undertaking because it was sort of the convergence of preparation and opportunity and circumstance, and taking advantage of it, not letting it slip away, was wise and I think valuable. In terms of medical genomics, we have a tremendous cost savings now because of technological advancements. And, you can not only sequence individuals, as you mentioned, but perhaps think about entire populations. I would imagine that someday this has the opportunity to radically change the future of healthcare. And we're starting to begin to see this. Can you give listeners a sense of the impact this could have?
Yeah, I think it really will change healthcare for everyone. To date, it has had a revolutionary effect for people with rare disease, and certainly for around half the families that manage to get to someone who can do genome sequencing for them. And that's really the first step, I think, on the way to the revolution that is genomic medicine. The impact of being able to sequence a genome cheaply and quickly and accurately for those who suffer from undiagnosed disease has been significant. I think because if you don't have a name, you don't have an understanding of what your disease is, of course, it's impossible to treat it. And although rare disease is actually collectively quite common, 1 in 10 people has a rare disease, so that still covers a significant portion of the population. Of course, I think the revolution is realized when a large majority of people are affected by something. And so I think now we're looking towards prevention, and essentially sometimes we would call that precision health, the idea of getting ahead of disease. And I think genomics has a large role to play there too, because I think understanding each person's individual risk for disease will really have to be incorporated into our ability and our approach to screening disease. At the moment, we do screening for, let's say, breast cancer or other forms of cancer based on age alone. That doesn't actually make a lot of sense because we know in the population there are people at much higher risk and some people at lower risk. And the same is the case for Parkinson's disease or Alzheimer's disease or other diseases for which we might do screening. So I think moving towards a personalized approach to healthcare, an individualized approach. I sometimes say that surely your doctor can individualize your care just as much as your hairdresser. Surely that's not too much to ask. We really have the tools to do it. And I think that the major tool is the human genome.
So, Euan, we listened to an interview you did with the New England Journal of Medicine's podcast where you talked about how someday we will see a pathogenic label or be able to order a pathogenic label on a genetic report. And a lot of people today are ordering genetic tests or working with genetic testing companies such as 23andMe, where people can learn, for example, whether they have the ApoE4 gene, which is related to an increased risk of Alzheimer's, or say a genetic variant associated with Parkinson's, or a genetic predisposition toward heart disease as examples. And it's remarkable what these genetic tests can tell us today. But on the podcast, you talk about how new, more advanced genetic reports are on the horizon and that they will provide even greater detail and will come with pathogenic labels. So can you talk about these future genetic reports and how they will differ from what we're seeing today?
Yeah, of course. I think it's a really interesting area and necessary for the future success of genomic medicine. The fact is that we all have variants. That's part of life. It's part of the great diversity of the world, and the fact that no two people, even identical twins, do not actually have identical genomes. We all carry multiple variants. And so, and as an example, I mentioned earlier, we compare every new genome that we sequence to the human reference sequence. In general, we will find around 4 million variants. So 4 million ways in which the person in front of us differs from that reference sequence. So that means that whenever we sequence a genome, that the problem is not going to be sort of searching for a needle in a haystack, but rather searching for a particular needle among, 4 million other needles. And that's a slightly different problem. And so when we think of a clinical genetic test report, what often comes back these days, either you may have a very clear variant that has been known or seen before, or just by nature of the way it kind of breaks a gene, it could be concluded just even the first time it's seen that it is likely to be pathogenic, that is contributing significantly to a disease. But there are many, many variants which are of lesser known significance. And we used to say, at least the entire genetics community would call these variants of unknown significance. I actually don't like that term at all, because these days we actually know something. We can say something about any variant. And that's because really of, in many ways, technology pushing us through. And you mentioned the population cohorts. So it used to be when we started doing genetic testing, if we would find a new variant in a particular gene, and then to know if it was new or not, we'd compare it to 100. I feel, amazing even saying this out loud, but, it's 100 anonymized blood donors. That was our population comparison. Well, now, without too much difficulty, you can put your hand on probably 1 million or more individuals of much more diverse ancestry. Back in the day, we didn't know anything about the ancestry, in order to make a comment about whether that's a variant that is common in the population or a variant that has never been seen before. And that's critical information. So I would say that we can get these next generation reports, first of all, by moving on from saying that just any variant in which we can't make a strong conclusion is of unknown significance. It is of some known significance because we have these population cohorts. But there's also really exciting new work. Our lab and others are involved in this, where we create essentially in cell culture every known variant in a given gene. And then we read out some phenotype or some change in the cells that we hope maps towards the disease that we're interested in all of those cells that we make the variants in. So we can basically make every single genetic variant. And they're called variant effect maps. And I think they will increasingly be available to allow us, when we see a variant for the first time, to say something useful about it. And finally, we have AI. And so just in the last month, we've seen from the Google genomics team something called alpha missense, which is really building on decades and more of work by many groups in thinking about, given a new variant and given all the knowledge we have of the world and how proteins work, and even thinking about comparative biology, other animals and their genomes, what is our best guess as to whether the variant in front of us would actually cause disease? And we've had those sorts of approaches for some time, but they've just been given an enormous boost by the power of artificial intelligence, and particularly our ability to model proteins through AlphaFold and other approaches, and these population cohorts. So I do think we're in a very different and very interesting world now when we come to interpret new genetic variants in patients for the first time.
You often compare the work of physicians and geneticists to detectives, because patients often present themselves as a sort of a medical mystery. Can you elaborate on this and talk about how you teach young physicians to think of themselves as a kind of a detective?
Yeah, of course. I mean, I think when we start, certainly if we talked about way back in my childhood, think about being a doctor, but if you think about how we think about doctors just in the world, we often think about not the everyday diseases where the answer is obvious. Someone comes to me and tells me they get chest pain after walking up a hill. They stop, it slowly goes away. It's a central crushing chest pain. We can all make that diagnosis. And if the first year medical student can't make that diagnosis, then they should be studying a little harder. That's a blockage in a coronary artery. But I think that's not really what we think of. And especially when you start to think about how doctors are sometimes depicted on TV or in culture and media. And it's not, I think, what drew many of us into medicine. What drew many of us into medicine was the idea of a medical mystery where the answer isn't clear. Someone presents with a syndrome or symptom or a series of symptoms that are unexplained. And they go to one doctor, maybe they go to another one, maybe there's some tests. And there's a remarkable number of people who live under the shadow of these symptoms and a disease with no name. And I think that we don't really understand until you were really there just how isolating that is. Many of our patients with undiagnosed disease talk about being on undiagnosed island, like literally on an island, separate. They don't have a community because their disease doesn't have a name. They don't know who to reach out to. They have nothing to Google. They have their symptoms, but they don't have the name of a disease. And of course, because they don't have a name of a disease, they don't have a therapy. And so it really becomes an area of medicine where we look to do both very traditional things, which is to say, really listen to the patient, think about their surroundings, think about their environment, think about their own biology, examine them very closely, bring experts in, and also apply technology. So something clearly I was attracted to for that reason. But when you think about that process, survey the scene, take in observations, think about it carefully, do sort of thought tests to see what it might be and what it might not be, run series of tests, come back and compare them to your hypotheses. That's exactly like what detectives do. I mean, if we're thinking back to some of the detectives in literature, like Sherlock Holmes, it's very similar. And when we think about how we teach young medical students, there's actually a lot of parallels between how we teach medical students to make medical diagnoses and how Sherlock Holmes might solve a crime.
So about 25 to 30 million Americans have a rare disease, which are sometimes referred to as mystery conditions. And you've been instrumental in establishing the Undiagnosed Disease Network, which was established in 2014. And it includes teams of physicians across the country who work with patients and their families to solve these medical mysteries. So I understand that the network has identified scores of previously unknown syndromes. And we're wondering if you'd be willing to talk about some of this work and also how the network first came about.
Yeah, of course. And this is really one of my favorite programs, and it really feels like a privilege to have been involved in it. The Undiagnosed Diseases program, which began at the National Institutes of Health, it really was the origin of the network. And this came about because of several individuals, but most notably a man called Bill Gall, who was a geneticist at the NIH Medical Center. And he'd been getting these rare conditions and just realized that in order to try to solve them, he needed a team of people. And initially, he got those teams together by kind of begging and borrowing resources, but eventually got a program at the NIH essentially devoted to undiagnosed diseases, funded through persistence and humor and charm and much else. But the good news is it was so successful that the NIH decided to essentially spread the love and turn it into a network. And so we were fortunate enough at Stanford to be part of that network, and I was really honored to be asked with Bill to co-chair the network as it came together. And the idea was simply to do what the Undiagnosed Diseases program as one center did, but do more of it and bring regional centers to those in need, because it was very clear that the need was much greater than the resources that Bill had in order to answer that need. As you mentioned, there are tens of millions of Americans, and one in 10 people essentially has a rare disease. And that's a lot. That makes rare disease collectively as common as diabetes. And so if you have a tool like the genome in particular that can be applied to most of those people, then it is reasonable to think of the people with rare disease collectively. But of course, they are very individual in a way that is very unusual. But one of the ways of solving that, in fact, it turns out our central tool, in 80% of cases, we're able to solve these mysteries by using the genome. So although we definitely take the Sherlock Holmes approach and we stroke our chins and think carefully and bring experts into the room and survey the scene, that maybe solves 1 in 10 of these cases. But of half the cases, in the end, our solve rate's about 50%. And so, which, if you think about it, for many of these individuals, they've gone for years going to multiple doctors, just accumulating masses of financial and emotional debt. And that's the moment they walk through our doors. So although 50%, I say that sometimes, and people look at this, oh, you must be disappointed in that. I mean, we want it to be better. But when you think about what these patients have gone through before they walk through our door, to be able to solve 50%, and most of those with one tool, the genome, I think that is rather remarkable. And so over the course of years now, we've solved hundreds of cases, we've established dozens of new syndromes. And really, it's all because of organization and dedication and our ability to understand the human genome.
Yeah, I think 50% is a remarkable number if you consider that some of the ailments may not be resolvable to the genome directly. In other words, probably that 50% is a very good performance.
That's very kind of you. We want to make it more. We want to do better. But you're right. We have to think beyond. Some will be environmental causes and may not be genetic at all.
Right.
STEM Talk is an educational service of the Florida Institute for Human and Machine Cognition, a not-for-profit research organization investigating a broad range of topics aimed at understanding and extending human cognition, locomotion, health span, resilience, and performance.
So let's talk about some of your recent research. Last year, you had a study that looked at the impact of genetic sequencing in critical care settings. Because a genetic diagnosis can improve the prognosis of critically ill patients and therefore guide the clinical management of the care, a lot of effort has gone into developing methods that result in rapid, reliable results. In a critical care situation, it seems obvious so that decisions need to be made in hours, not days. However, traditional testing often requires weeks, and even rapid testing requires days. Your paper that was published in the New England Journal of Medicine reported how you and your colleagues developed a method for rapid sequencing of the whole human genome in patients in as little as 5 hours. This new ultra-rapid genome sequencing has the potential to lead to significantly faster diagnosis. For this study, you used a technique called Nanopore Sequencing. First, can you explain for our listeners what Nanopore Sequencing is and a little bit about how it works and the advantages associated with it?
Yeah, of course. And sequencing the human genome is basically about spelling out every letter, which essentially means we have two copies of each gene and 2 chromosomes, one from each parent. So we're looking to give us the most complete picture of that. And what we do currently, so before nanopore sequencing and before long read approaches to sequencing, most of the genome sequenced to date have come using a procedure called sequencing by synthesis, which is a short read approach. Basically, we blast the DNA that we usually derive from your blood with some ultrasound to put it into small segments that are like something like 150 bases, or letters long. And then we essentially grow on these little clusters the mirror image of each individual strand. And then those clusters, as each base is added, are read off. That's the traditional way we do it. Nanopore sequencing works a bit differently. And a nanopore is a protein with a hole in it, basically. Not in too much more. It is more complicated than that, but that's a way to think about it. And so instead of building a DNA, which is how many of the sequencing approaches to date have worked, we sort of unzip the DNA so that a single strand falls through that hole. In fact, it falls through really quickly. And so actually you need a little motor protein to slow it down. And so this has been developed, a technology that was invented in California and pushed forward there, but has really been developed by Oxford Nanopore, a company in Oxford in the UK. And essentially, if you multiplex those pores, so you put hundreds and thousands of them in a membrane and pass a current through the membrane, then as a molecule falls through, and at this point we're talking about DNA, but it could be RNA, and in fact people are now looking at sequencing proteins this way. But as that molecule falls through, then there's a change in that current, and that current can be detected. And then using essentially these days an artificial intelligence approach, you can take the squiggle, and it is literally called a squigglegram. So imagine a very badly written signature, that's the output. But you can then feed that into a neural network that will translate that into an A, T, G, or C. And in fact, it looks at several bases at once as the strand of DNA falls through the pore. And if you multiplex that enough, if you have enough of that happening, you can start to sequence really quite quickly.
So can you tell us about the sequence of one of the study's participants that was completed in just 5 hours and 2 minutes? And it set the Guinness World Record for the fastest DNA sequencing, which, by the way, we didn't actually know there was such a category. So there must be a good story behind this. So please tell us, how did you end up in the Guinness record book?
Yeah, that's not every day that happens. Well, first of all, huge credit to those on whose shoulders we stand. Back in the day, when we first started doing genome sequencing, it became clear that there was a category of patients in whom it was important not only to have a genome that was cheap enough and accurate enough, but also a genome that was fast enough. So sometimes you need the data quickly, and we were standing on the shoulders of others, and in particular, a colleague of mine, Stephen Kingsmore, had been starting to think about how to deliver genomic data quickly. At the time, he was in Kansas. He's more recently at Rady Children's in San Diego. And he had essentially managed to sequence and interpret a genome in 56 hours. And that was really impressive. We thought that by tweaking the computational pipeline a little, we might be able to go a little bit faster. And for a couple of our cases, we had managed that. But then he jumped ahead and basically managed to sequence and interpret a genome in 26 hours. And at that time, he actually got a Guinness World Record for fastest genomic diagnosis, which was fascinating and amazing. And much more importantly, he started to apply this technology broadly so that many little babies and little children were able to benefit from getting genomic information rapidly. He also demonstrated this was highly cost effective. But of course, we were interested in whether it might be possible to go even faster again. And of course, we're not competitive, are we? But it was a really interesting question as to whether nanopore sequencing could actually allow us to go faster. So there was this machine that Oxford Nanopore produced called a Prometheon, a very well-named machine. It's almost like an entire sequencing center that sits on your desktop. And it has 48 flow cells, and individually within each of those cells, there are thousands of channels and individually probably something like 500,000 nanopores. And so if you think about 500,000 of those pores with DNA flowing through them and pushing data out, then you could cover an entire genome once in just 1 or 2 minutes. I mean, just mind-blowingly fast. Now, if we want to make a genetic diagnosis, we're going to have to do it more than once or twice. We'll have to cover the genome something like 50 or 60 times. But suddenly we're talking about the possibility of sequencing a genome in maybe as little as an hour or an hour and a half. And so we thought, well, what if we brought that technology to the table? Then what do we have to do next? Well, we had to think through every single element of the genome sequencing, including, how fast could we get the sample from the patient to the lab? And, the PhD student who was in charge of this program actually was an ultra distance runner. So we used to joke that he would literally run it from the hospital to the lab, which he actually did on occasion. And then we thought about how to derive the DNA quickly, how to get the DNA ready to go in the sequencer quickly. And then on the other end, the biggest roadblock was, well, how are we going to analyze this data this fast? And so the computer that came with the sequencing machine that was designed to do the analysis, well, if we put this much data, we fired this much data into that computer, it took 18 hours to analyze it. So clearly, if we were trying to beat a record that was at the time somewhere around 19 hours, that wasn't going to work. So we needed a new plan. And basically, another PhD student started working on a way of piping the data into the cloud rapidly and then running the analyses in real time. And we worked with other collaborators at Baylor College of Medicine, at Google, at NVIDIA, and then, of course, with Oxford Nanopore. It was really an incredible group of people coming together, almost a bit like that first genome. It was really kind of exciting moment to see, could we do this faster? And could we get that data then back to the bedside team to make decisions that really matter for critical care patients? And so, yeah, I mean, just to give an example, one of the patients, Matthew, who's been happy for us to share his story, he was first winter of COVID, presented with a cough to his local hospital in Oregon. Turned out he didn't have COVID. He had severe heart failure and was going into shock. So he was airlifted to Stanford, where it was very clear that his organs were beginning to fail. And so we put him on a heart-lung machine just to keep him alive, and then implanted an artificial heart device just to keep his organs functioning. The question was, how did his heart go from essentially being normal to completely failing? And there's at least a few different possibilities, most of which have been ruled out, but we were left with perhaps one of them being myocarditis, so an infection of the heart muscle, which would ultimately potentially be reversible, or a genetic heart condition, which almost by definition would not be reversible. So the question was, how would we differentiate the two? And even a biopsy of his heart muscle, where we go in and take a piece of the heart muscle and look at it under the microscope, that didn't resolve the question. So we enrolled him in our study, and basically just 8 hours later, we were able to make a diagnosis of a genetic heart condition, finding a variant in the troponin T gene that explained his underlying dilated cardiomyopathy. Because of that information, the team was able to make the decision to put him on the transplant list, and he actually received a new heart 2 weeks later. And among many amazing things in his story, they sent a regular test at the time, regular genetic test, because this was a new thing. Of course, we sent the regular test. It didn't come back till 3 weeks after he received his heart transplant.
Yeah.
So I think that just being able to get this information fast can allow people in those critical conditions to get answers and make decisions quickly to help patients. That's who it's for at the end of the day.
Absolutely. And I further understand that you and others at Stanford want to cut the current fastest sequencing time roughly in half. If that's true, that ambition is true, how far off would you guesstimate that is?
Yeah, I have said that, haven't I? It's like setting unrealistic goals for my team. We're aiming in that direction. We have actually already managed to beat the record we set that you mentioned earlier, and so we have already been able to go faster. And we are starting to look at every single place we can shave off another few minutes. I think being able to do it in half the time is going to be a big lift, but I think that we can get there. And of course, it raises the question of how fast is fast enough? But if you're talking about critical care, illness, and you're talking about sudden death and people literally patients whose hearts are stopped, then I don't think we can go fast enough. And that's part of what I tell the team. I think we have to decide. The other thing is that this approach, because it's long read and long read sequencing, is we're not going to sacrifice accuracy. It is possible to go faster if you're willing to sacrifice accuracy. But this is somebody's life we're dealing with. This is clinical genetics, and so there's a minimum bar for how much genetic information and of what type that we want. But that tension is always there. If we slow down, we can usually squeeze out more accuracy. So that's kind of where the team is playing. As I mentioned, we've shaved off a bit, and it may take us a year or two though to get to half.
So you recently published a paper in Nature that introduced Cosmos, which is a cloud-enabled platform that performs real-time cell imaging and analysis. So Cosmos is an acronym for computational sorting and mapping of single cells. And in your paper, you describe Cosmos as a novel platform based on AI and microfluidics, and it's capable of high-throughput imaging that can sort cells using deep morphological assessment. So cell morphology has been used by pathologists and also clinicians for years as the gold standard for disease diagnosis and prognosis. And although there have been technological advances in making single-cell characterization at the genomic, transcriptomic, and also proteomic levels, tools for assessing high-dimension cell morphology have not kept pace with advancements in molecular characterization. So really exciting. And can you talk about the challenges that you and others have faced in performing real-time deep learning assessment and sorting of cells, and also how Cosmos helps address these challenges?
Yeah, of course. And, it's an amazing thing with the world has moved towards single cell biology, and that's really exciting. We have these tools, some of which we've talked about here, genomics, of course, and transcriptomics, looking at the RNA, proteomics, thinking about the protein complement, metabolomics, thinking about metabolism. All of these have been downsized to the point that they can actually be applied at the single cell level. But one of the amazing things to us, and this is something that occurred to us a few years ago, is that we make all these high dimensional measurements, so millions and millions of measurements. But imagine if we did this with humans, we do it without really looking at the cells. So imagine if we were looking at a population of humans and we sequenced their genome and we looked at the transcriptome of their white blood cells and we looked at all their proteins, but no one stopped to see, well, how tall was that person, or how much did they weigh, or, just tell me some basic things about them. And it's just really an artifact of the moment that we are in time. As you mentioned, the history of morphology and thinking about cells and their shape and size and how that changes with function has traditionally been the realm of pathologists. And pathologists, by definition, look at dead tissue. It's embedded and it's dead and it's cross-linked, and you look at it under a microscope, deep and dark in the middle of the night. But new technology allows us to keep those cells alive and separate them into individual cells and then potentially flow them in small microchannels. We can even rotate them a little in those channels and then take pictures of them. And we can do that at scale, and we can do that at high resolution. And if we think about then this massive leap forward in our ability of neural networks, like deep learning architectures just in the last 10 years, to be able to recognize objects in the world. Could be your dog. We can recognize a dog from its ear in the corner of a photograph, or a frisbee on a beach. I mean, it's not that long ago when that seemed really miraculous that a computer program could do that. Now we are, as of this year, in the next revolution from AI of large language models. And so the deep learning mode of just a few years ago seems like old now. That's last year's revolution. But the fact is it hadn't actually reached everywhere. And one of the places that AI revolution was taking its time to reach was single cell biology. And so we really were impressed with the combination of the move forward in artificial intelligence approaches, single cell microfluidics, and then the ability to understand biology at a deep level. And so the Cosmos system really is about bringing those things together. And a very talented postdoc in my lab, Madison Museli, worked on this and felt that there was really an opportunity to build a system. But there were a large number of challenges to doing that. It's not a trivial thing at all. In particular, being able to track a single cell at speed in a channel and then sort it, push it in one direction on the basis of, let's say, a deep learning algorithm is a very non-trivial task. So we started a company out of the lab called Deep Cell, also with Mayar Salek, three co-founders, to really try to solve this problem. And the team has been growing over the course of the last few years and has been putting this in people's hands, most recently, this new technology. And we're really excited by what the community will be able to do with it in adding morphology to these very high-dimensional genomic measurements.
Have you thought about where you'd like to go with future enhancements of Cosmos? We've talked about AI and how that might be used in more advanced ways with respect to cell biology. Could you talk about how this might lead to new insights that could have significant clinical and translational implications?
Yeah, of course. I mean, I think one of the drivers for us in thinking about this at the beginning was not just to think about what the scientific community might do with the tool, but rather thinking about the circulation. The circulation is a remarkable thing. It's basically a biopsy of every organ in your body. I mean, your bloodstream goes, think of a red blood cell in its average day. It's circulating around the body. It's reflecting the health and disease of every organ, of the bloodstream is of every organ in the body. And so taking a blood sample is a remarkable thing. And I think we've only scratched the surface of what we could do with that blood sample. And in recent times, we have started to look for cell-free DNA or RNA, which is a new world, and that has brought many revolutions in terms of thinking about early detection of cancer or non-invasive fetal diagnosis. But what about, I think, what about the rest of the organs in the body? We know for sure that many of them are leaking cells into the circulation. Also, why do we wait till somebody is ill before we start to look for these things? And so I do think that there's a new world where we start to look in people's circulation for rare cells, and we can use this technology to do that. And so that rather than taking a biopsy, let's say of your liver, if you have liver fibrosis, we could look in your circulation. We can pull out those liver cells, and we can do all of those omics technologies that we just talked about to those cells. And they're alive, so we can culture them. So I think there's a really interesting and important future of health disease prevention by really exploiting the circulation at the cellular level much more than we have to date.
It's as an aside, Euan, it's been fascinating watching the application of the evolving, and not always improving in my view, but the evolving AI technology as it's applied to medicine. In the right hands, it's been splendid. And in the wrong hands, you see people using AI without understanding the limitation of the particular technique they're using.
I think that's absolutely true.
It's very interesting indeed.
The paper that we just set off, I think last week, was partly, I think you'd have enjoyed it because we realized that for the ECG, understanding ECG, some of these new AI models were just better. Like the F1 statistic was, maybe it was 92 or something, but we can get to 89 with a linear regression with 5 terms in it. It's like, okay, so you get, a tiny bit more, but with this huge computational cost and this black box nature, as opposed to 5 easily determined, easily definable features that anyone in the world could read from the ECG. And so
less opaque.
Right.
Yeah,
that's exactly right.
Opaque.
Yeah.
So Euan, returning to Steve and the work that you did after he showed you his sequenced genome, you mentioned earlier how Steve pointed out to you a variant in one of his genes associated with heart disease. And it's a variant that very possibly could be life-threatening. And accurate assessment of cardiac function is crucial for diagnosing cardiovascular disease. In a recent paper of yours, you address the limitations of human assessment of cardiac function. To overcome this challenge, you and your colleagues have developed a video-based learning algorithm, which is called EcoNet Dynamic. And this surpasses human observation in several critical tasks that are related to assessment of cardiac health. You reported on this development in a paper that was titled Video-Based AI for Beat-to-Beat Assessment of Cardiac Function. So what went into the development of EcoNet Dynamic, and also what makes it stand out as an assessment of cardiac function?
Yeah, well, we've been spending a lot of time thinking recently about the ways that artificial intelligence and neural nets can really help us. We're focused on augmenting the physician and making the sum of both better than the individual parts. But of course, there are going to be places where the artificial intelligence can be much better than a human, and there's going to be places where that will not be the case. I mean, going back to an earlier point of our conversation, the thing that first drew me into the heart was that it moved. It's the only organ in constant motion. And, that was one of the challenges, because when we first thought about applying those algorithms that we talked about earlier that can recognize your dog or a frisbee or whatever on the beach, a mountain, then the first thought, of course, in medicine was to apply those to static images like chest x-rays and CT scans. But of course, it had occurred to us that it's just another step towards video. And of course, as the tech world moved from static images to video and the application of neural networks, then we knew that leap was something we could also apply in medicine. And we're better than the heart, of course. So if we think about how we look at a heart, our standard tool is ultrasound. So we shine the ultrasound at the heart and we see it moving. Depending on which view we take, we can see 3 chambers or 2 chambers or 4. But one of the most important elements that we try to derive from an ultrasound of the heart is how well is the heart essentially squeezing? How much blood does it squeeze out per beat? And we express that in a fraction known as the ejection fraction, so the fraction of blood pushed out every beat. And we realized that is currently being measured in thousands of places around the world by individuals literally taking one frame from one video and then tracing around the inside of the heart and then scrolling forward a few frames to the point where the heart's at maximum contraction, and doing exactly the same thing again, and then calculating those areas and taking them apart. That is not a great use of humans' time. And of course, if you can only really do it once, but a computer can be trained to do it not just once, but many, many times. And so this was the idea behind the EchoNet system, is to be able to derive ejection fractions automatically from ultrasounds so that they could then be reviewed by a cardiologist and approved, but just to save humans time, essentially. And it does more than just save time, because it allows you to do multiple measurements, which makes your measurements more accurate and more precise. And it means that especially in situations where you have, for example, a rhythm like atrial fibrillation, where your heart is beating very irregularly and each beat is a slightly different shape and size, whereas before you would just pick an example one, now with EchoNet Dynamic, you can actually look across tens or really as many beats as you want and average them and give you a much better and more precise estimate of heart function. So that was the original idea. And another important part of it was that the world only gets better by sharing data. And so we were able, and huge credit to David Ouyang and the team. David led this work at our lab, and James Sobe was my co-senior author on this, that we really were able to get to the point where we could share 10,000 of these ultrasound videos with the world. And now on any given week, there are people around the world working on that data to do better. And that's, I think, a really interesting element of the tech world that is now coming into the medical world, where as long as we can appropriately anonymize and de-identify data, we can share it broadly and let really anyone work on it to make it better.
Here at IHMC, we have three primary overlapping research thrusts. The first in original area was AI, and the second that joined up here was robotics and exoskeletons, and the third, more recent area, is health span resilience and performance. And in fact, we're building a new research complex dedicated to health span resilience and performance research. Thus, I was really interested in your paper published in Nature last year, The Genetics of Human Performance. As you pointed out in your article, although we have substantial epidemiological evidence supporting the beneficial effects of exercise, we really don't know much about the molecular mechanisms through which these effects operate. We do know, however, that exercise seems to extend health span. In this article, you reviewed the current understanding of the genetics of human performance, and it was an excellent article. Can you talk about this and why it matters in terms of modern humans who have become largely sedentary, unfortunately?
Yeah, of course. And first of all, a shout out to Dan Kim, who was the cardiology fellow who led this work and led this paper. We've had an interest in human performance for many years. I just think, we spend so much of our time dealing with the frailest in our world and those with the weakest hearts. I mean, we're a big heart transplant program here at Stanford. But in terms of understanding the heart, I think you can learn a lot, of course, from the weakest hearts. But I think there's a lot you can learn also from the strongest hearts. And we've had a real interest in elite human performance and what we can learn about the fittest in our society in order to help the frailest in our society. And so that has led to a study that's been going on for many years called ELITE, where we're sequencing the genomes of the fittest people on the planet. But I think that there's this, just as you say, there's this really interesting kind of evolutionary component to heart disease. Through most of our history as humans, we've been much more active than we have now, and we've had a completely different diet, a completely different approach to eating food that has led us with a particular genetic complement that allowed our survival over many hundreds of thousands of years that is mismatched with a world where we currently live, where there are elevators and escalators and McDonald's on every corner. And I think that that's something that isn't often appreciated, but is something that is really critical to trying to reduce the prevalence of heart disease. If you look at modern hunter-gatherers as our sort of best window into the world of more ancient hunter-gathering populations, then certainly they would be walking 5 or 10 miles a day. There are many of us who don't go anywhere near that in our current sedentary world. And they would be eating a combination of foods that could be foraged, but also every now and again, there would be this opportunity to do what I think most accept as the strategy of earlier humans to hunt through persistence hunting. It is one of the advantages we have, that because we're upright in 2 legs and we have bodies that are not covered with hair, that we can thermoregulate much more effectively than most quadrupeds. And so although we're definitely not the fastest runners, certainly not over a short distance, if we're able to keep the prey in sight, then ancient humans are able to continually follow that prey until it gets to the point of exhaustion because of our better thermal regulation ability and because of our aerobic potential. And so I think that when we think about that and we think about the genetic complement that allowed our survival under those kind of conditions, it is not well suited to our current environment. And so we do, I think, have to understand, if we're trying to have humans be more healthy in the modern world, that we have this complement of this kind of baggage of our evolutionary history, if you like, that leads us to want to eat high fat, especially saturated fat and highly dense caloric foods. And overall save energy, because it would be important to save energy because you don't know when your next big meal is coming from. We have those predispositions in a modern world, and what we end up with is obesity and diabetes and the challenges that we see around us.
So you and your co-authors reviewed the large body of research that has shown that all forms of exercise, and in particular endurance exercise, confer benefits across just all different types of domains of health and function. And you then went on to review recent research that identifies specific genetic pathways that may underline the beneficial effects of endurance exercise. So can you talk about these genetic pathways and also the key points of your review, and as well as your primary conclusions?
Yes, of course. And I think there are many elements to fitness as we think about it. And one of our interests in elite athletes is that really every element of physiology, if you think about the analogy of a car, I mean, every element has to be performing at its absolute highest potential for that car to go as fast as it possibly can. And it's the same thing with the human body. And so when we think about the different specific genetic pathways that count, when we think about aerobic exercise and VO2 max in particular, human performance is often quantified using the maximum amount of oxygen that the body can consume at maximal exercise. Then we think about different systems. You have to be able to pool in oxygen very efficiently. You have to be able to make sure that oxygen gets in and stored in your blood very efficiently. You have to be able to transport it around your body efficiently. And then you have to extract it from the circulation and get it to the exercising tissues. And so there have been leaps forward over the course of many years in each of those different aspects of the delivery system. Of course, you have to also then take the metabolites from exercise away from the muscle. And that's an extremely important element too. So initially, these studies were very underpowered, and it was very hard to make too many conclusions. But because of really a large community effort, we've started to identify common pathways, some metabolic pathways, mitochondrial pathways, thinking about the heart, the pathways that allow the heart to operate at high efficiency, pulmonary pathways, and circulatory pathways as well. So obviously we go into a lot more detail in the paper, but really just thinking in a systems way is what I think has led the community to move forward in our understanding of the genetics of human performance.
I'm always a little cautious when people use the word fitness. We all kind of know what's meant, but when you think about it, fitness for what? Fitness to be an NFL lineman is not much like fitness to be an accountant. It's not much like fitness to run a marathon, right? So It's one of those words we all use. I use it, but somehow it without the context, it's less meaningful than one would like. That was by the way, I really liked that paper. And speaking of pathways, in the paper, in one of the figures, the little text by one of the figures, the conversion of lactate into propionate in the gut, which is, if taken up by skeletal muscle cells, may contribute to enhanced physical activity via AMPK activation. This is really an interesting point. What are your thoughts on the significance of the role the microbiome may be playing in enhanced endurance, physical activity in general?
Yeah, I think this was just one of the most fascinating papers I've maybe ever read, because it was surprising, and the case was made, I think, very clearly by these authors. I mean, we have really under-recognized the role of the microbiome in, I think, life. Most of the cells that we think of as us are not actually human, right? They're bacterial. And also when we think of diet, anything that we ingest through our mouth is going to meet the microbiome first before it meets any human element of our body. And I think that when I finally realized that, and it took me way too long, it was just a moment of revelation for me. And papers like this were among the ones that opened my eyes, because who would have thought that the bacteria in your gut were going to be part of your exercise metabolism? You think of rest and relaxation. So the time, you think of the systems shutting down when you're eating and digesting, and that being the opposite of high intensity endurance exercise. So we really think of the gut as something that isn't really playing a role. We think of fight or flight as what we're doing when we're exercising. So this paper was really revelatory. This Villanella species that is capable of converting lactic into propionic. It's just, a sort of insane thing. I would never have believed it if someone had said. But they showed very elegantly through a variety of experiments in this paper, including a microbiome transplant at one point into other animals, that this really was true. And I think it's just one of many papers that have linked the microbiome to a number of different things, even psychological changes in a mouse's ability to fear heights. They show that a microbiome transplant can change that. So I think there's a whole new world for us to really begin to explore through genomics and metabolomics of what is really happening with our gut microbiome and how it affects our entire world.
Agreed. We've had several microbiome researchers on STEM Talk, including Alicio Fasano and others, and it's so complex and really fascinating. I only found that paper because you referenced it in your paper in the little note, next to the figure.
Yeah, I don't think we didn't have room to really go into it in detail, but to me, it was such a fascinating finding that I wanted to make sure we included it somewhere. So I think Dan eventually, he squeezed it in, of course, I think just in a figure legend.
So while some genetic variants identified to date account for small portions of the variance in exercise training adaptations or human performance more generally, it seems the major determinants of inter-individual differences may lie in dynamic expression responses, which are largely influenced by dynamic epigenome. What, in your mind, are the critical paths forward in exercise epigenomics?
Well, understanding, of course, essentially the anatomy of the genome, what is different in the A's, T's, G's, and C's is in many ways just the beginning, because it's the expression of the genes and the parts, the regulatory elements in between those genes that really determine our biology. And so a major part of that, of course, is epigenomics. And it's certainly been known since the earliest days of our ability to quantify the epigenome that exercise is a powerful modulator of that. And our tools are only getting better. I mean, up till recently, we've had to do special techniques to be able to read the epigenome. Think about methylation of the genome, for example. But nanopore sequencing, like we were talking about earlier, or the sequencing from Pacific Biosciences, these long-read approaches are actually able to interpret the epigenome and read out the epigenome using just the same raw signal, but just a different algorithm, which is somewhat mind-blowing. So we're now reaching a point where we can sequence the if you like, the anatomy of the genome, but also read out some genomic function at the same time. And I think that's going to be, incredibly important path forward as we sort of upgrade our approach to sequencing, so that we just roll in at least this particular element of epigenomics to our approach. And we're part of a consortium called the Molecular Transducers of Physical Activity Consortium, where we really try to think about those molecular transducers of both strength training and aerobic training, initially in an animal model. And that data is actually already available with a paper coming out in the next few months. And the human study is ongoing, which is one of the largest studies of its kind. And I know one that Marcus Baumann, who you've had on the podcast in recent times, is a major part of. And so I think we're looking forward to be able to answer questions like this about exercise epigenomics using that resource over the course of the next few months and years.
Yes, Marcus is leading a large part of our health span resilience and performance effort here. And in fact, Marcus volunteered this next question for you. The previous question notwithstanding, you are involved in projects via the WuSci Human Performance Alliance to identify new genetic variants that have larger effect sizes on human performance than those previously identified. Can you talk about the importance of the ELITE project in this context?
Yeah, of course. And I think it goes back to our fascination with this idea of an engine working at its full potential. I think we can learn from those who are the weakest in our society and whose organs are failing, but there's so much that we can learn from those who operate at the elite end and the people who have the highest, let's say, VO2 max or the highest strength, and those who can leap the highest and run the fastest. And so we started a project many years ago. It was inspired by the story of Eero Mäntyranta, the famous cross-country skier who had lived under somewhat of a shadow of blood doping, because every time his hemoglobin was measured, it was really high. But eventually it was found out that actually he and his family members suffered from a genetic condition that basically left the accelerator down on the bone marrow. So he was making more red blood cells. This was essentially a genetic genetic doping, but it wasn't his fault. He was cleared in the end. But that idea that we could identify ways, and that pathway, of course, is now a drug target. So we use erythropoietin to be able to boost the red blood cell production for individuals who have anemia from a variety of causes. And so it's a great example, we felt, of the way that we could study elite athletes and yet learn something that really could help a much broader group, including those with disease. And that's the essence of the ELITE project. So we're recruiting, if you know anyone or any listener thinks they may be in that elite category, and it's not necessarily, you don't need to have an Olympic gold medal. In fact, we have some Olympic gold medal winners who actually don't reach the criteria to join the study, but we also have plenty of people who have never been particularly competitive, but find out that they're extremely fit from a VO2 max perspective. And we're interested in their biology, and particularly we're starting with the genome. And so you can actually, it's a global study. We're recruiting from across the world. And these days, of course, many people have watches, digital devices that estimate their VO2 max. And although they're not perfect, those estimates are actually not too bad. And so if anyone out there is finding their watches estimating their VO2 max is above 55 for women or 65 for men, then we'd love for them to get in touch, and we will sequence their genome, share the results, and try to learn about the superhumans in our society.
So you certainly have a full plate, and somehow you managed to find the time to write the Genome Odyssey, which the Wall Street Journal describes as an impassioned firsthand account of the effort to bring genomic data into clinical practice. So in writing the book, what were you hoping to accomplish?
Well, I hope you've had a sense just in the time we've had to chat today of just the privilege I feel being in the middle of this, first of all, amazing place at Stanford, but also in the stories of these patients who are afflicted with these rare and undiagnosed diseases. I see what they go through, and I compare it to my very privileged life, and I just feel like I wanted to try to tell some of those stories, and so that's what happened. I had no idea whether I was capable of writing a book. I still, at some level, don't believe I really did, but I at least started with the idea of trying to tell their stories in the best way I could. And an agent and a publisher were happy enough to come along for the ride, and yeah, I was really happy to see that come out. And it's even nicer to get stories from people who have read the book from around the world and have shared that it resonated with them, or in one case, someone told me they got a job because they read the book. I don't believe that, but at least it's helping people, I think, understand. Well, I hope the wonder of the science that we've talked about here over the last period, but also really what these families go through and what the genome has been able to offer them in terms of answers to these really difficult questions.
So in the Wall Street Journal review that I just mentioned, it highlights your account of the remarkable progress of genetic medicine over the past 2 decades, and then also mentions how you believe more wondrous advances are on the way. So could you talk a little bit about some of these advances?
Yeah, I think, and we've touched on some of them a little in this conversation. I mean, I think the genome is coming cheaper and faster and better. But I think that we're also going to start integrating the genomic data with data that we can get from other places, like we just a moment ago mentioned digital and wearables. I think that we're really looking to try to move from a reactive healthcare system to a proactive healthcare system where we get ahead of disease and we can implement prevention rather than treatment. And when we think about the ways of doing that, we talked about precision medicine. I think it's not just measuring the genome. I obviously believe the genome is a huge part of what we can do with precision medicine. But think about what we can do if you understood that maybe you were particularly at risk from your genome of a particular heart rhythm. And then maybe that is known by your watch that sits on your wrist and actually monitors your heart rhythm. And it alters the pre-probability of making that diagnosis in you based on your known predisposition. I think when we start to integrate those worlds, we have a really interesting situation coming about. Or another example might be Parkinson's disease. If you think about the particular shuffling gait that goes along with that, or the pill rolling tremor that some patients have, again, we have digital devices that can pick these up early. That data should be talking to the data in your genome to understand how likely it is that person who's on whose wrist the watch is likely to suffer from those conditions. So I do think that we're moving forward into an interesting world where we really can achieve this and realize this goal of preventive care.
So one last question for you. In your Stanford bio, it mentions that you're a father of 3 and that in your spare time, and I laugh when we think about that, you're very busy, as mentioned, that you play jazz saxophone, you pilot small planes, and you conduct research on the health benefits of single malt Scotch whiskey. And I will tell you that I'm dying to know what the health benefits of single malt Scotch are.
Well, hopefully they're obvious. It tastes really good. I obviously come from Scotland, and so from from an early age, with colds were treated in Scotland with something called a hot toddy, which involved a small amount of single malts. Maybe it wasn't single malt Scotch. So it's certainly something that we've had an interest in for a while. I've enjoyed many moments of both scientific discussion and medical discussion over a glass of single malt Scotch. Hopefully lower blood pressure, more relaxed psyche, and great realizing and hatching schemes of great ambition are probably among the health benefits of single malt Scotch.
That's awesome.
Your bio also says you're on a quest to understand American football. Now, how is that coming?
Yeah, well, that's a hard one. I mean, the human genome is pretty easy, but understanding American football is a whole other challenge.
Or the fans.
Yes, exactly. I'm still on the quest. I still hope to understand more. I was brought up on obviously soccer, the football that you hit with, where you play the ball with your foot, and rugby, where at the moment the Rugby World Cup is on. So I take that bias into my understanding. I'm always wondering in American football why they just don't lateral the ball more, because that's how rugby works.
Yeah.
But that's maybe a discussion for another day.
Different podcast.
Yeah.
Well, Euan, this has been great talking to you. Thank you so much again for joining us on STEM Talk today.
Oh, thank you so much for having me. It's been really fun.
It's been great. Thank you, Euan.
STEM Talk.
STEM Talk.
STEM Talk.
STEM Talk. STEM Talk.
So, Ken, we so often hear people talk about how they're conducting pioneering research and how they're on the cusp of a new frontier. But definitely have to say, Euan certainly is a pioneer, and his work really does promise to radically transform healthcare. And that was just a really fascinating and fantastic interview.
Yes, Don, you're right. Euan is certainly at the forefront of the application of genomic data to clinical practice. For listeners who are interested in learning more about Euan and this important work, be sure to check out his book, The Genome Odyssey.
If you enjoyed this interview as much as Ken and I did, we invite you to visit the STEM Talk webpage where you can find the show notes for this and other episodes at stemtalk.us. This is Dawn Carnegie signing off for now.
And this is Ken Ford saying goodbye until we meet again on STEM Talk.
Thank you for listening to STEM Talk. We want this podcast to be discovered by others, so please take a minute to go to iTunes to rate the podcast and perhaps even write a review. More information about this and other episodes can be found at our website, stemtalk.us. There, you can also find more information about the guests we interview.